I'm Sneha Bheemireddy, a graduate student from India. I'm very thrilled to share my work with all of you. We have titled our work as Understanding the Significance of Interprotein Bifurcated Interactions in Protein Complexes. I'm very happy and thankful for all the authors who have worked with me to achieve this. Now, let me introduce you all to this project. I'm sure you must have come across one or the other protein complexes in your work. Now, imagine there are three protein chains in a complex, and let's say chain A is interacting with chain B and C. Or, in the given example, as you see in the slide, chain A is interacting with chain C, chain E. Now, what do you think will be the prevalence or occurrence of these interactions among all the protein complexes? That's what made us curious and took this question, thinking what would be their prevalence? If so, what would be their significance? With this curiosity, we have shaped our objective as understanding the occurrence and significance of these bifurcated interactions across all multi-protein assemblies with known structures of high resolution. Before going any further, I would like to introduce you or clear about what do I mean by bifurcated interactions here. Bifurcated interactions are those interactions where a residue from chain A is simultaneously interacting with residue from other two chains. For example, now residue isoleucine 187 is interacting with isoleucine 187 from chain E, again isoleucine 187 from chain C. So now the green color residue from chain A can be called as RBI, residue involved in bifurcated interactions. These two residues, they can be called as participating RBI, the residues which are involved in participation of the bifurcated interactions. With this motto, let me go further to tell you about the steps we took to study our objective. Now, as usual, we began with dataset preparation, where we took all the protein structures from PDBs with resolution better than 2 angstroms. Then we identified all the interprotein bifurcated residues using our in-house server PIC. We have classified these interactions into hydrophobic, electrostatic, hydrogen bonding, van der Waals, etc. We looked at the propensity of the amino acids involved in it along with the frequency of the interactions. Then we identified hotspot residue to understand or study their significance towards stability of the interfaces. We consider a residue to be a hotspot residue when, upon mutation to alanine, it changes delta delta G higher than 2 kilocalorie per mole. We went on to calculate the conservation score where we used conserve, and we also calculated the precise interaction energy between two atoms using INTAA. This led us to believe some results where we realized that arginine and leucine are the amino acids with high propensity to be RBIs, and majority of the interactions are contributed from hydrophobic interactions. Although our study is novel, there is one previous study which has similar work, and the link is given below. Now, let me give you a detailed analysis of our results. We also saw that every protein complex has only one type of amino acid as RBI. As you have previously seen, that entire protein complex has only isoleucines as RBIs. That is very interesting. Again, we have looked at the nature of the interfaces and we see that 51% are partially obligate, 32% are obligate, and 17% are non-obligate interfaces. In general, any protein interface prefers leucine, which is followed by arginine, and generally has least preference for sulfur-containing amino acids. Whereas participating RBIs have high preference for non-polar residues than RBIs except for methionine. We also realize that they occur as pockets across the interface. Then we further went on to see what would be the prominent interaction, and we realized that it would be hydrophobic interactions followed by salt bridges. We have also looked at the prevalence of hotspot residues and their frequencies, and we see that hotspot residues also reflect this trend as hydrophobic interactions are observed to be high in frequencies than other amino acids like leucine, isoleucine, tyrosine, and arginine. We further went on to see the conservation patterns of interfacial residues, and we realized that these are very similar to conservation patterns of participating residues. For example, in the figure, A depicts the conservation patterns of the interfacial residues, whereas C depicts the conservation patterns of participating residues, and we see that A and C are very highly similar. Whereas B depicts the conservation patterns of RBIs, which are residues involved in bifurcated interactions, and we see that arginine is the most conserved. If we have to look at the amino acid pairs, among the most favorable amino acid pairs, it would be cysteine arginine and cysteine methionine. With this, we'd like to take you back to the conclusions. And before going further, I would like to say in this work, we have highlighted the significance of residues involved in bifurcation and their contribution to the protein complexes. To conclude this talk, we have realized that arginine and leucine are the amino acids with high propensity to be RBIs, and hydrophobic interactions play a very significant role in bifurcated interactions, and tyrosine contributes very significantly to the stability, followed by proline and cysteines to be involved in bifurcated interactions, which were very highly conserved. Thank you so much for listening to my talk, and I would like to take an opportunity to acknowledge Professor N. Srinivasan, who is no longer with us but has guided us through the project, and Professor R. Saudamini, who is overlooking this project now, and Revati Menon, with whom I have shared this project. This would be me, and I would like to thank all the current and past members of NS Lab, CSIR, IISc, NCBS, and ISO Tirupati for their funding, and last but not the least, organizers of ISMB 2022 for giving me this golden opportunity. Thank you, everyone.